So, good morning, everybody. Today we are going to cover Chapter 11. Chapter 11 is DNA technology or the topic of biotechnology. So, there are a lot of advances that are happening in biotechnology, and that's where the future jobs are. So, DNA technology refers to manipulating DNA. So, scientists, they can manipulate DNA in different ways. They can change the sequence, and because we already did the DNA replication, transcription, and translation, you know that if the sequence in the DNA is changed, it's gonna change the sequence here, and then it's gonna be expressing a different protein. So scientists, they can maybe cut out some of the letters, and they can put different letters there. They can switch off the genes, so they can switch them on. That's called epigenetics. That's a field of working with the genes, and they can transfer DNA between species. They can take, maybe they like this one gene in one species, and they want the other species to have the same gene. They can take out that gene, you can insert it in the other species. And then usually they do this for plants to get a higher yield for the crop and to decrease the cost for the herbicides and the sprays that the farmers need in order to, get rid of the insects that might be harming the plants. So they can take a gene for a toxin that makes them herbicide resistant, and they can insert it in the crop. Now, when the farmer sprays the field with the herbicide, the plants, nothing happens to them, but the insects die. So to advance agriculture, they can manipulate the DNA of the plants or the crops. And then for the crime scenes, if there is something found at the crime scene and they want to see who the person is, they can go ahead and copy the DNA, make multiple copies so they have enough to kind of identify it, and they can use it like that. So first thing I want you guys to know is the definition of a transgenic organism. Trans means to transfer, genic means genes. Transgenic organism have genes from another organism, where the genes have been transferred into this organism from another organism. And that DNA that this organism has is called recombinant. It's been recombined or spliced and then put together. So recombinant DNA is genetic material that have been spliced together from multiple sources. It can be from 3 different organisms. It can be from 4, it can be from 2. So I really need you guys to know the definition of a transgenic organism. That's an individual or that receives recombinant DNA. Then you have to know what recombinant DNA is. Recombinant DNA is recombined from different sources. So it's not coming from one. Have you guys heard about glowfish? Fish that glows? Yes. Nancy, what have you heard about it?
Is it like golden fish?
It's not, oh, Nancy said she has one. So it's been, so at first it was used as a hobby in the household. So this fish, it's found in Ganges River in India. And they took a gene from a jellyfish and they put it in this fish. And that gene was for a glow protein. And now this fish glows. Another thing they were able to find out that this fish glows when there are certain chemicals in the water. So they use this fish to detect pollution. So some estrogenic chemicals, the glow responds to that in the reproductive system. So they can use this fish to detect pollution in water or detect certain chemicals in water. But this fish would be an example of a transgenic organism. And this fish has a recombinant DNA. In this case, I said it was a jellyfish gene. So they took a jellyfish gene and put it in this fish. Another example is Bt cotton bollworm. So Bt cotton, Bt stands for Bacillus. It's a bacteria, Bacillus thuringiensis. You don't have to know the name, but it stands for that bacteria name. So this is cotton. and it's still kind of growing. And then there is this insect that's infecting this cotton. So what they did, they took the gene from the bacillus bacteria. Bacillus bacteria produces this toxin, and they put it in this cotton. Now this insect cannot infect the cotton anymore because it has the toxin. If it would bite on it, it'd probably die. So that's how they were able to get a lot more yield for the cotton plants, and it also helped economically and financially. So that's another example of transgenic organism. There is transgenic bacteria, and transgenic bacteria would be a bacteria that has a gene from another organism. They can take bacteria now, and they can take the insulin gene in humans. So people who have diabetes, maybe they cannot make insulin, they need to inject that in their body. And to make that insulin, they can use bacteria. They can take the insulin gene and they can insert it in the bacteria. Now this bacteria is going to be producing insulin, and that's how they're able to produce lots of insulin. Also some blood clotting factors and fertility hormones. Those are just some examples. And then they can also change some stuff in bacteria, where bacteria can degrade petroleum, pesticides, and other soil pollutants that wouldn't be degraded otherwise. So they can manipulate the genome for the bacteria. They can insert things that can change the function, and they can express different proteins. The way they do it, and they use plasmids. So they use plasmids to create transgenic organism. And hopefully by now you already know what transgenic organism is. So a plasmid, for a definition, I want you to remember that it's a small circular double-stranded DNA. It's not part of the chromosome. It's found in bacteria, but it's not part of bacterial chromosome. It's just a small circular DNA that's just floating around on the side, but it's not part of the chromosome. But this small circular DNA, it's able to replicate itself inside the host cell. So it's able to replicate, and it can be used to express the genes that that plasmid might have. So if I ask you what are plasmids, you're gonna say it's a small circular double-stranded DNA. It's not part of the bacterial chromosome, and it can replicate itself. So scientists, they use plasmids to manipulate the genes. So I want you guys to remember how this is done. I'm going to go over that right now. If I'm spending more than a few minutes on a slide, it's definitely going to be on the exam. So just make sure you know the steps that I'm about to go over after. And if there are any questions after, please ask. So to begin making a transgenic organism, they are going to combine a plasmid with the source DNA. Source DNA would be something that they're interested in, a gene that they're interested in. And then they cut out that gene that they're interested in with something called restriction enzymes. Restriction enzymes in biology are known as molecular scissors. It can cut enzymes. So they cut it out. The restriction enzymes cut out genes, actually. Sorry, I said enzymes. They cut it out, and then they put it in the plasmid. So that's what's happening here. They have the DNA they're interested in, and they're on the DNA, maybe there's a gene, they cut it out, and there's a circular double-stranded DNA plasmid. They insert it in the plasmid, and then they put the plasmid in the bacteria. So here it says that you have to have the DNA you're interested in, you have to have a plasmid, you cut out the gene, and you cut the plasmid and you put the gene in. And then after that, maybe there's bacteria that they have in this test tube. So there's maybe bacteria that they have in this test tube. They're going to put all the plasmids in, and then they're going to shock the cells, bacterial cells, with electricity or with heat shock, or they can just use the gene guns and they can just take this plasmid and shoot it inside the bacteria directly. Also, you guys remember endocytosis and exocytosis. That's when the membrane bubble forms around, it just fuses with the cell membrane, and that can be done too. Or they can just use a virus that infects the bacteria. They can take the viral DNA out, and they can put the plasmid in, and the virus can go ahead and infect this bacteria. And then this bacteria is going to have the new gene that they were trying to put in this gene. So you have the recombinant plasmid here, and now this bacteria is transgenic bacterium that contains the human DNA. So I'm going to draw this out for you. So this was a summary. So I'm going to draw it out for you. And if there are any questions, please ask. And participate, maybe help me out. So this is your double-stranded DNA. So I'm going to say DS for double-stranded, then I'm going to say DNA. And then there is a gene on this DNA that you're interested in. Maybe this is the gene. What is the first thing you need to do? So maybe type it in the chat or say it. So what is the first thing that you do?
Separate it?
You separate it. How do you separate it? So how do you separate this gene from the DNA? This is the gene you're interested in.
The restriction is an enzyme.
So you cut it using, so maybe you cut it here using restriction enzymes. And I must say cut. So now you have this gene, and now you can obtain a plasmid, which is a circular DNA. So you can get a plasmid. You need to take this gene and you need to paste it there. So you want to paste this gene there. And sometimes there are overhangs. So you see there's an overhang right here. There's an overhang right here. So that's little breaks that are in between this gene and the plasmid. Do you guys remember from DNA replication, if there are any breaks in between, how do you fix it? There's an enzyme that fixes it.
Ligase.
Ligase. The ligase can fix these breaks. And then, so you cut this gene, and then what you did here, I'm gonna say paste. So cut and paste. Paste it in the plasmid. That's what you did. And then ligase helps with the pasting. So it just helps with the pasting. So there are no breaks after. Now you have that. Now you need to have bacteria that you want to put the plasmid in. So I'm going to draw a test tube. This is your test tube. And these dots are bacteria. And usually they use E. coli. So I'm going to say E. coli bacteria. So you take this plasmid, and not just one, multiple. There's a lot of bacteria there, and you put it in the test tube. Now, what do you do so the bacteria can take the plasmid? There are different methods you can do. What do you do? Give me one.
Shock them?
So you can shock it, zapping with electricity, or you can heat shock it. You can do either or. So once you do that, some of these bacteria, they're going to end up taking the plasmid. So then you take the bacteria, you put it on a Petri dish. I'm gonna draw a Petri dish. And then this is your, maybe this is the plasmid. So let me draw the bacteria first. And then you have your bacterial chromosome right here. You already know plasmids are not part of the bacterial chromosome. You have this plasmid with your gene of interest. So some of these bacteria are going to look like this. They end up taking the plasmid. This is bacteria with plasmid. But I said not all of them do that. So first thing you do after you have your bacteria with the plasmid, you're going to put it on a Petri dish. This is your Petri dish. And then you put the bacteria on there. You have your nutrients in the Petri dish. So I'm gonna say nutrients. Now, this is your bacteria. And I said, not all of the bacteria take up the plasmid. So maybe this one took up the plasmid. So this one is, good to go. This one didn't take up the plasmid. So you don't want that one. This one maybe also didn't take the plasmid. You don't want that one either. So what do you do to determine which ones took up the plasmids and which ones didn't take the plasmid? So when you're putting this gene in, your gene of interest, you can also put in a gene for antibiotic resistant. So you can insert this gene in as well at the same time when you do this. Then what do you do in your Petri dish with the nutrients? You can add some antibiotics. Now the bacteria that took up the plasmid has also taken up the gene for the antibiotic resistance. When you add these antibodies to the Petri dish, then the bacteria that has taken up the plasmid survives because it's also resistant to this antibiotics. And then the bacteria that did not take up the plasmid dies. So that's how you can differentiate. So you're only gonna be left with the bacteria that took up the plasmid. Any questions so far? Yes, Nancy. Do you have a question? No
I
questions?
think I have.
Okay, George, go ahead.
So you have like on the nutrients assays, you have like three circles and you marked two, kind of no, then the one good. I don't really get that section.
Okay, so here's your bacteria that took up the plasmid. So when I said you put in the plasmids with the bacteria, not all the bacteria pick up that plasmid, even when you heat shock it. So this one I put check here. I'm trying to say maybe this is the bacteria that taken up the plasmid. I'm not drawing the whole bacteria here. I'm just drawing a circle for this whole thing. Then this is a bacteria that didn't take up the plasmid, and this is the bacteria that also didn't take up the plasmid. So you don't want this bacteria. You only want the one that has taken up the plasmid. So that's why I have three circles here. I'm just trying to give you examples, not all of the bacteria that you have. So how do you determine, which one has taken up the plasmid? And that's when I said you add a gene here for antibiotic resistance. So the bacteria that takes up the plasmid also takes up the gene for antibiotic resistance. And once you add the antibiotics, this bacteria is going to die because they didn't pick up the plasmid. And this one lives because it picked up the plasmid and the antibiotic resistant gene with that. Does that make sense, George?
100%. Thank you.
So make sure you know the steps. Again, I don't give you guys essay questions, so you don't have to write it from scratch. It's going to be one of multiple choices, but make sure you know the steps. Then there's one more thing. This can be done to plants as well. And the plasmids that are used for plants are called TI plasmids. TI stands for tumor inducing. So if you see a TI plasmid, immediately I want you to think about plants. So TI plasmids combine with plant chromosome, and TI plasmids usually have a gene that infects the plants with tumor, but we can modify it and we can insert a different gene, in this case herbicide resistant gene, and when this TI plasmid goes in the plant, now that plant becomes herbicide resistant. So TI plasmids are tumor inducing plasmids used for plants. So once this plasmid enters the host cell, in this case plant cell, the cell can express the gene carried by the plasmid. And you can put any gene in this plasmid. In this example, again, it's showing the bacterial gene for herbicide resistant. And then this we already went over. The fish expresses a jellyfish gene for glowing color. This is a transgenic organism. This is a transgenic organism. They have the recombinant DNA. Recombinant DNA can be put in using TI plasmids here, and then the other plasmids here. Also, I said the viruses can be used for the transgenic animals. So what they do, they take the viruses they remove the viral DNA from the virus, and they can infect or either early embryonic cells with the virus, or maybe um, animal can inhale the viruses depending on whatever they're working with. So if they infect the early embryonic cells, then the cells divide by mitosis, like it usually do, and then growth happens, development happens, but this DNA gets passed on because it was in the embryo when it was inserted. So to all the animals, so it gets passed on.